Hello everyone, my name is Nagarjun. So I'm working as a scientist, like a research and development scientist at ANSERMA. So today I'm going to give you a talk about like introduction to the next generation bioanalytic development. So I would like to focus like on this title slide with the image right there. So the image itself is kind of like showing you the complexity of like from a small molecule to kind of like going into a recombinant protein, which is like an antibody, and then you go for like a virus, which is like a gene therapy product, and then you go for a cell, which is kind of a cell therapy product. So it's kind of the complexity that increases from a small molecule, chemical molecule, and you jump into a biologic. As the biologic increases, you just have to understand how much complex these drug products are. So I just want to start my presentation by giving a confidentiality statement here. So most of if the presentation will be talking about like the current literature, and there may be forward-looking statements in them. And again, this is just based on the current literature that's been published. And again, no assurance that like these expectations or estimates will be achieved or kind of like will be accurate in future. So the overview of the talk would be I'm going to really introduce to the cell and gene therapy products right now in the market, and then I'm going to talk about like a drug product life cycle, including how analytical development uh, is involved or like is being attached into this drug product life cycle, and then talk more about like critical quality attributes and on a general sense, and then I would talk more about critical quality attributes in cell and gene therapy. This talk mostly focuses on gene therapy products, so I will talk about gene therapy a lot. And then I would talk about different analytical methods. What are the traditional methods as well as like advanced methods that are being applied to define these kind of like critical quality attributes in cell and gene therapy products. And I would give one case study, and which is a article, and then I will give final thoughts of like how these critical quality attributes can be kind of like understood or achieved in a gene therapy product. So as an introduction, I want to really talk about like the cell and gene therapy products. And before that, I would love to introduce like what is like a gene therapy and a cell therapy. So the one on the left is kind of more towards to the gene therapy product where we call it as a direct delivery. So in which you kind of have your transgene or a therapeutic transgene, and you kind of embed that into a kind of a viral genome, and which is kind of like non-immunogenic viruses. So there are different kinds of viral vectors people use, which is kind of more related to like adenoviruses, adeno-associated viruses, and lentiviruses. There are also retroviruses people have been using. And once this product has been produced, it's been introduced intravenously or specifically tropism into the region of therapeutic area, which can be applied. The one on the right is kind of like a cell therapy product. So they are kind of like they can be divided into two things. So cell therapy product can be a stem cell that can be differentiated into kind of like the required cell type, and that can be like delivered to a human, kind of given to a human for a specific disease. Or another way people also create cell therapy products is kind of like you take this viral or non-viral delivery and deliver the specific transgene into a specific cell, and then you kind of like manufacture them to high quality and then provide them to kind of the human. And most of the raw material that's coming for cell therapy is kind of like when you consider a CAR T therapy is the blood itself from a patient, and you kind of like engineer it in such a way that like you give back to the patient for kind of like overcome the kind of the disease. So there are kind of like 9 products for cell therapy. Kymriah and Escata are kind of like the ones that have been approved previously, and there are 7 more products that have been approved. And for gene therapy, there are kind of like 2 products approved. I'm going to talk in the next slide. What are they? And it's predicted that like there will be more than 20 to 30 gene and cell therapy products will be coming through by 2030. So for the gene therapy approved therapies, there are two gene therapy approved therapies. The first one is the Luxturna, which is kind of an adeno-associated virus 2, which is primarily focused towards the inherited retinal diseases. And this has been approved in 2017. And ideally, what the gene delivers is like an RP65 gene, which has been delivered into the retinal region of the eye, and it kind of like enhances the vision. And the other one, the other product that's been approved by FDA in 2019 was the Zunzma, which is the AAV9, and which is primarily focused towards the smooth muscular atrophy disease, and in which you kind of give the SN1 gene, which kind of has like an exon 7 or 8 being deleted in a diseased patient, where you kind of give that exon to make a functional product with a gene therapy using a gene therapy product. So I would like to talk more on the drug product life cycle and analytical development here. So when you look at like a drug product life cycle from a development stage, so there will be an early stage development, and there'll be a late stage development, and there'll be a commercial development, and these will be subdivided based on the product life cycle. Would be mostly all the preclinical and the phase one would come under the early stage development, whereas the late stage includes the phase two, phase three. Once you go to commercial, you're like talking more about the approval and the market. You can see the same life cycle on a manufacturing scale. So most of the time, a research and development really finds an ideal lead candidate, and that once it's taken into like a process development where the engineering GMP is developed, where you come kind of like do all your engineering runs, and that's mostly till where you complete your phase one, phase two, and once you're done, you are kind of like transfer into like clinical GMP manufacturing, which is more towards the phase three. And once you kind of like get the drug approved, and it really moves the drug into like commercial manufacturing, and you're kind of like have very specific sites for this commercial manufacturing. And you can look at the same life cycle from analytical development, where as these drug products are being developed, you always have to have assays to really define this drug product. So most of the design, define, and evaluate and optimize of assays really happens during the research and development process. And once you have like an assay which is kind of like more defined and have all your controls ready, you kind of like do your qualification, which kind of stands around that phase one, phase two. And once you're moving that into like phase three, the assay moves into phase three, it has to be validated, where it's kind of like more specific regulations in validation, and you kind of like test all these parameters for validation of the assay. And then once the drug gets approval into a market, it's the assay itself is moved into commercial manufacturing kind of setup, and it's called like it has a lot more verification of the assay and robustness and all other things have been tested. So again, so this is kind of like just giving an overview or landscape of like how things happen. So there are a lot of bottlenecks in viral vector manufacturing right now. Sorry. So the key areas where the bottlenecks really happen is kind of like which the key areas where R&D and kind of the CMC has to come together to really meet this commercial stage demand for this kind of like viral vector and really produce like a more safety product. And the key areas where R&D or the CMC focuses a lot with the current state of like cell and gene therapy products is kind of like the first bottleneck would be the lack of optimal production system. So what it really means is like we don't have kind of like so all these viral vectors that are being manufactured is through a transfection process most of the time, where you have like high grade plasmids being like transfected into some kind of a cell line. A simple example would be like a triple transfection of adeno-associated viruses, where you're giving three plasmids into a HEK293 cells to produce our virus out. So the biggest challenge with this process is kind of like when you kind of start dealing with like multiple steps for production, it's not always like robust, and the variation is very high from lot to lot production, and which kind of like delays most of the production, or like you have to do multiple lots to get like enough amount of product. So there are few kind of like places where people have been working. So one of the like area where people have been putting more time or investment into is kind of really developing a producer cell lines for this like viral vectors, which again recombinant protein is a good example where CHO cells have been like modified or engineered for production of these antibodies, and which is kind of using a producer cell line, and they get a very consistent lot to lot uh, production. So another area where people are investing a lot is kind of like it takes a lot of time for producing this producer cell lines. So people are also investing in synthetic biology to really understand, can we engineer kind of like the cells in such a way that like where you can reduce where you can understand the metabolic system of a cell, you can modify in such a way that like you can enhance the viral protein production for in such a way that like you'll have like more of this viral vector being produced per scale. So another big challenge is kind of the product potency, where you kind of have like a product that's been produced for a specific lot, and it's not very consistent, and it may be where you have a high requirement where you're not really achieving that. In that case, you are kind of like consistently pooling lots to create that high amount of dose. So again, that this is still like the characterization of the potency assays have to be like worked out, and that's where the field is also working on. And another area is kind of like the non-adaptable use of this upstream bioprocessing. So most of the time, as the demand is so high, most of the companies, or like most of the time, we kind of like work with kind of the transfection model and carry out the whole engineering run. But once it's been transferred into a kind of like the initial commercial or kind of like the phase three manufacturing, you kind of have to have a more robust process, and as the scale is like it should be scalable. So whatever the upstream bioprocesses right now are not really scalable. That's a big challenge or bottleneck. Another area is kind of like the absence of very suitable analytics and properly defined critical quality attributes for a cell and a gene therapy product. So this is kind of like like an important area where the critical quality attributes are not really defined, but there are like investments happening in such a way to really define these. So I'm going to talk more about the critical quality attributes. So looking at critical quality attribute from an FDA guidance point of view, so a critical quality attribute can be defined as a quality attribute that should have an appropriate range. It should be a specific limit for it, and or the distribution in such a way that like it will ensure the product quality. So somehow very critically define the quality of the product. So like in a general scale, like in a general guidance, like how are these critical quality attributes being defined when you consider like a small molecule world or like a recombinant protein world? So once you have like a lead candidate and you kind of like want to really understand uh, a critical quality attribute for that lead candidate, so what normally people start with is kind of like they kind of start with a quality target product profile. So it really taking the drug product and really understanding, oh, what is the mechanism of action of this drug product, and what is the efficacy and safety limit, really understanding that, and what's the dosage form, is it really given like in a liquid form or any other different form, and route of administration, is it like taken given like intravenously or intramuscularly. Once all these are defined and they really come together and fit into regulatory requirements, once they start really taking the product and they start looking at the quality attributes. So these quality attributes are really defined by two important factors. One factor is called severity, and the other factor is called likelihood. So severity is kind of like depends on the safety and efficacy of the product. So the severity kind of takes into account the kind of like when I say safety, is this drug product safe enough to give to like a human? That's the first question they'll be asking. And when they talk about like efficacy, is this drug product efficacious enough? Like whatever product that's been produced has good amount of potent product that's been able to create that efficacy. And they also look at the likelihood. So the likelihood is defined based on like what is the chance this drug product can actually cause a adverse event. So they kind of give scores based on that one, and they calculate a casualty score using the severity and the likelihood. And once they give a casualty score for all these quality attributes, they kind of rank them and identify the ones that are kind of like a particularly specific quality attributes. And they kind of define more like a control strategy around it, really understand, deep dive into these quality attributes. Are they really defining the product as during a process? So when I say process, it can be a kind of like a manufacturing process. It can be a preclinical study or it can be a clinical study. So as they get all the information, they start doing kind of a risk assessment. And for the risk assessment, it's kind of a repetitive process to really understand these quality attributes, and then they finalize like this critical quality attribute identification. So this is kind of a regular process that happens for small molecule as well as the recombinant protein. So, but based on some of the initial data, the field for cell and gene therapy actually provided a critical quality attributes uh, definition. So, the critical quality attributes for cell and gene are primarily taken from the recombinant protein world and been applied into cell and gene therapy products. So, this this table actually defines kind of like few elements for a critical quality attribute. So one is kind of attribute class. So which is kind of like when you take a drug product, it should have some specific identity. It has to have a purity, safety, potency, and quality. So for a cell and gene therapy product, we kind of use different methods to define these attribute class, and each class will have a quality attribute. So if I consider identity, if I take a gene therapy product and if I'm using a genomic method or a proteomic method, I should be able to give a quality attribute, a specific attribute. It would be if I'm using an a adeno-associated virus, I should be able to give using my methods. So this is the transgene, which is very specific, and this is the phenotype, which may be the serotype of the specific virus. It may be like adeno-associated virus 2, 9, and the genotype will give you the specific transgene you want to deliver. So this is kind of like a quality attribute. So we also have guidance for this from FDA. Again, these guidances are all still from the recombinant protein world and then applied to cell and gene therapy. So as we look at this table, so there are five different attributes. And when you look at these test methods, there are multiple analytical methods have been applied to achieve a attribute class. So as you look at like purity, it's primarily focused around the product and process impurities, where you kind of understand, oh, how is your viral vector aggregation happening, or there are other product impurities that are coming with the drug product itself. So these are like one another example. So we can also look into kind of like each attribute class and different methods, analytical methods that have been traditionally applied. And as we want to understand the next generation bioanalytics here, I would give like an kind of like an more overview of like how these different class attributes have been identified using a traditional method and an advanced method. So the first attribute class we can look at is potency. So which is kind of like a very important attribute class for viral vectors and cell therapy products. So potency actually defines the kind of like the efficacy of the product. So we are kind of an example would be a viral titer for viral vectors we can talk about. So a viral vector of a can be like in traditional method normally people use is kind of like a quantitative PCR. So in traditional method, the quantitative PCR it's kind of have like some of the drawbacks. One of the drawback would be it's like low reproducibility. The reason why is kind of like you're always in need of a qualified standard, and the standard itself, manufacturing the standard itself, can create that reproducibility issue. So the second would be people have used on a protein technology is the ELISA. So the ELISA is kind of like more like a time consuming and it has a lot of assay variability. And you ideally have to design specific assays for every serotype dependent. So you have to have like an ELISA for AAV2 or AAV9. It's very specific and you have to really optimize it to your specific serotype. It's also time consuming. So as the cell and gene therapy products are complex, the people have identified like applying advanced methods would be a kind of like an orthogonal way of really applying advanced methods would be ideal to define these products better. So people have been using traditional methods and now people are like slowly moving into these advanced methods, and for genomics people started to use the digital PCR. The reason why it's like more sensitive on, so you can have like a low high range on the lower end where you can identify like very low copy numbers, and another thing is like it's an absolute, so you really don't need a qualified standard for that. When you want to use a proteomic method, people have started using the size exclusion HPLC methods. The only reason is kind of like high throughput. You can run like multiple samples, and again, based on it's kind of like a more sensitive and like a low variability is like very low because like it's based on like you read the peaks, you have control. So it's kind of like so these methods have been started to apply for this initial potency assays to define uh, kind of like more of these quality attributes. So the next quality attribute we can look at is the purity. So when I say purity, it's kind of more focusing towards the impurities and the purity of the drug product. So the impurities can come from the process as well as the product impurities. So the traditional methods people have been using previously, and still people use these traditional methods. Is kind of like one is like the STS page, which is kind of like a very low sensor, and like you need a large sample requirement. And ELISA, as I talked, is like more time consuming, and assay variability is very high. And this another method where people also use is the um, capillary ultraphoresis, which is kind of using a UV sensor, which is again has that low sensitivity. And to kind of like advance and identify purity, so the field itself had started using these advanced methods. One is to kind of for the genomics, people have started using the next-gen sequencing, where which is kind of like highly characterizable and highly sensitive. And the other one is the mass spectroscopy, where you have like you can have a high throughput and highly sensitive, and like you get a huge amount of data about your drug product from mass spectroscopy. Another like a quick method people have started using the capillary electrophoresis with the kind of like the fluorescence fluorescence detector. So where the fluorescence detector itself will give like a high sensitivity, and a small sample volume is required to really achieve identify like kind of the quality attribute as a purity from this method. So the other quality attribute for cell and gene is kind of the identity, which is kind of like really saying, am I really producing the right drug product here? So for that, the traditional methods are kind of like Western blot and the capillary electrophoresis. And Western blot itself is like has low sensitivity. You have to have like a large sample volume. You have to have a specific uh, concentration to really identify your viral vector. And as like an advanced method, you have like people started to use like mass spectroscopy, so it will give you like a more better characterization information, and it will give you like more about like how is this product itself, like, or what is the modifications does the product have, as these been produced in mammalian cells, these viral vectors, are there any post translational modifications happening, and will give you like a lot more information. And also the capillary electrophoresis with the fluorescence detector also is kind of like very quick and gives you more kind of like requests more information. So the the other quality attribute would be the drug qualities, product quality, in which where we are kind of like looking at it's kind of like, oh, you have a drug product, final quality. Oh, is it you purified the whole viral vector product or drug product? What do we have in there? Is there like really a potent product, or do we have like, you know, any non-specific drug product being available? So for the traditional methods people have used is to really kind of like once you I didn't want to really get a very pure product. People have you started people use in traditional methods is the analytical ultracentrifugation, which is kind of very low sensitivity and it requires really large volume. And another simple kind of like a traditional method is the optical density, which is kind of using the 260 by 2 or like 260 for DNA and 280 for protein, and really getting like an understanding of like the product quality. Again, it's kind of like a low sensitive and kind of like again requires a larger volume if you're using a cuvette actually. So the more advanced methods with the product quality, because like product quality has become like a more uh, kind of like as you're really looking at the final product that goes into like a preclinical study or a clinical study, people have put more effort on really understanding the product quality here. And again, the advanced method people started to apply is the anion, kind of like the anion exchange chromatography with HPLC, so which kind of gives you uh, very specific kind of like sorting the sample out and really high throughput and highly sensitive, and kind of like gives you a better characterization. And the other one people start like the literature stated, or like most of the groups started to use, is kind of this multiple detectors attaching to HPLC. So they're kind of like using different detectors on HPLC to really define this product quality. So it really gives like more information on an orthogonal way. It gives you accurate and robust information, and it also measures like multiple physical attributes at the same time, and it's also highly sensitive. So I think like as the field evolves, I think this is where the field will be evolving more, as because these are complex products, you really have to identify like a number of things around the drug product. So maybe this detector application will be like will be seen more. So on the same lines, I can actually give you a one case study actually. So in this study from Gangans, what they did was kind of like they kind of used a multiple monitor HPLC study to really identify in-process monitoring. So they've been producing like a V production and they're purifying the AAV. So there are multiple steps in the process. So they want to use this HPLC to really use kind of like, okay, a normal HPLC comes with a UV detector. They want to add like this in kind of the kind of the light scattering, eccentric, and the intrinsic fluorescence detectors. And then they want to really identify is are they able to really give them enough information about the drug product, about the purity, and all of the critical attributes. So the key takeaways was kind of like when they used like an intrinsic fluorescence, they were able to really detect like the sensitivity of AV detection was more compared to UV. When they kind of used kind of a light scattering, uh, they were able to really identify AV capsid in complex samples. So as the sample is being purified, it starts with this cellular lysate, and you kind of like slowly reduce the cellular lysate and get your drug product out. And that's what they're claiming, like the light scattering was able to really identify even in a complex samples. And the other, when they use like an extrinsic fluorescence, they were able to really track all this the DNA from the host cell, and they were able to really show them like when you use all these kind of like uh, monitoring systems at one time, the complex samples like gene therapy samples were able to be clearly provide good answers about the product. So this the kind of like what this graph actually shows, the chromatogram show here, is kind of like they were showing data kind of with the UV, where you look at like they kind of show like it's only like 18% of like empty capsids. When they read the same sample with the light scattering, uh, kind of like a light scattering uh, diode, and what they found was it's like around 24% of empty capsids. And when they use an intrinsic fluorescence, they were able to show like around like that 26% empty cap. So what they want to really claim is kind of like the UV detector, which traditionally used, is kind of like more kind of like underestimating. Uh, it's just because like the reason why is like UV just uses like tryptophan to give the reading out. So there's a high possibility that like it can be or underestimated sometimes. So and that's what they claim, like once they start using multiple kind of like the monitor sensors, they were able to really define in a more clear way. So another, again, the article, I just took like very few figures. There are like more data in this article, but like this is just an example where they did an experiment where they kind of like took the cellular lysate and applied like proteinase K. So they're trying to reduce like all the histone and all the protein on the background. So they kind of couple the UV with the extrinsic fluorescence using the pico green. So pico green specifically is intercalating dye, goes and really interacts with the DNA. So when you have like a double stranded DNA, this dye can really sit in the double stranded DNA. So using an UV, what they're showing is like the UV itself is not able to pick any chromatin in the chromatogram, the first chromatogram. And when they add like a proteinase K treatment for 1 hour and 24 hours, what they're showing is the green is kind of the pico green. So the pico green completely shows it's become high sensitive and really picks all the plasmid and the DNA that's present in the sample, where it's clearly showing all the protein is being chewed off by the proteinase K. But when you look at the UV, which is the blue kind of like the protein, and the kind of the red is kind of like your DNA, the UV is not so sensitive enough to really pick enough amount of DNA. So yeah, so it's clearly showing that like once you kind of have multiple kind of like monitoring systems, it clearly shows like it's becoming more sensitive and accurate for a complex product like a gene therapy product. And I speculate like these kind of studies will happen more and really will be like this is where the next generation bioanalytics for cell and gene will be like moving into. So so as I said, like we talked a lot about like critical quality attributes. There are multiple challenges to apply traditional application of critical quality attributes for cell and gene therapy product. So the schema I showed you before, it's kind of been defined for small molecule and recombinant protein, and people have applied that schema and really were able to identify the critical quality attributes easily for uh, for other products. But the challenges that can come for this cell and gene therapy product when you're trying to identify these critical quality attributes will be like the first challenge would be because there are a lot of products in research and development, and very few products have been approved by market, and lot has been in clinical trials. It's like very limited knowledge. We just have very limited knowledge from the clinical point of view to really identify these critical quality attributes and risk assessment. And it's again the batch production tends to be limited. As I said, like there's a lot of batch to batch variation when you produce these. So one batch may be very potent, the other batch may be not potent. So these are like a lot of the variability itself is a limitation to really identify the critical quality attribute. And there is not much of similar products to really accumulate enough of like this risk assessment data from clinical trials to really show that like okay, oh, this is a product this which may be similar. I can really identify critical quality attribute. A simple example would be you have an antibody and you create a biosimilar. So you have like data from clinicals on the antibody and you create a biosimilar. You kind of like take the data and really compare, oh, what can be the adverse events that are coming in. That's a simple example I can tell. And the other would be the product development knowledge is very limited. It's just like we are still understanding how are these produced and what's the best way to get the right yields. And again, as I said, the knowledge is limited is based on like how the production manufacturing is still not really up to the scale as we expected. So placing the same schema, how can we develop like a critical quality attribute identification in gene therapy? So I took the same schema that's been applied to like a recombinant protein. So once you define the quality target product profile, so you kind of come and define your quality attributes. So the quality attributes are actually defined in the table below based on the product related and the process related. So normally, so there are different models people apply. And for gene therapy product, we should go with the preliminary hazard model. The reason why we go with this model to identify critical quality attributes is kind of like the preliminary hazard model actually takes into consideration only the knowledge that's been available on the product, limited knowledge that's available on the product. And most of the IHC guidelines, when you want to identify critical quality attributes, request to get data from clinical trials for the risk assessment and then apply for the quality attributes and define the critical quality attributes. So as we don't have enough of kind of like scientific literature or the data, so the preliminary hazard model takes into consideration that aspect, and then we can apply that model to really identify our critical quality attributes. So yeah, so it's again the schema is the same schema and kind of like the product and the process related quality attributes, and we start really the next step would be really taking each of these attributes and calculating kind of your severity score and the likelihood scores, and then you kind of come up with the criticalities. Uh, you come up with the criticality score, and then you kind of rank them, and then you go apply through the process and really define your critical quality attribute. And once you have your identified critical quality attribute, then you start doing, you use your design space quality by design and apply to that. And so I have some of the references for the talk, which kind of like have uh, again, these are kind of like more related to like analytical methods and process and drug characterization, and again, new the latest drug develop, bioanalytical processes being applied. Yeah, and thank you very much for the time.